My name is Ruwaida Al Khalil. I'm an international board certified lactation consultant and registered nurse. The objectives are mainly to describe the mechanisms of drug transfer into breast milk, but as a prerequisite, I will describe briefly the pathways of milk secretion and synthesis by the mammary epithelial cell. Now, the use of medication is one of the major reasons why women stops breastfeeding prematurely. As such, it could be because women they fear harming their baby because they are using drugs, or if healthcare workers they don't have enough knowledge about the transfer of drug into breast milk. So, in order to promote and maintain breastfeeding, it's very important for healthcare workers, not just only lactation consultants, to know about mechanism of drug transfer into breast milk. One takeaway is that the percentage of drug reaching the baby from the maternal medication is less than 1% for most medication. Now, in coming videos, I'm going to talk in details about maternal medication groups, and I'm going to go through specific medication and to discuss the relative infant dose in them. But in general, it's almost most medication have less than 1%. Now, regarding the first objective, to briefly describe the pathway of milk secretion and synthesis by the mammary epithelial cells, there are mainly 5 pathways in which substances, including some medication, pass through the breast milk. The first pathway is exocytosis of milk proteins, lactose, and other components of the aqueous phase of Golgi-derived secretory vesicles. And you can see here, these are secretory vesicles that throw up their contents into the milk ducts. The second pathway is milk fat secretion via milk fat globules, and as you can see, the cytoplasmic lipid droplets bulges out from the cell cytoplasm to form the milk fat globule, which can contain medication in its center and in its membrane. Pathway 3, direct movement on monovalent ions across the apical membrane of the cells. And pathway 4 is transcytosis of components of the interstitial space. And pathway 5 is the paracellular pathway for plasma components and leukocytes. Usually, pathway 5 is open at the initiation of lactation, at the beginning, cholesterol phase, then it closes later, and it might reopen during pregnancy, involution, and when there is inflammation in the breast. And this part is going to be discussed in future videos that talk about formation of human milk and biochemistry. Now, when we talk about the mechanism of drug transfer into breast milk, the mechanism of drug transfer into breast milk are no different than those governing passage into any other maternal fluid or organ system. Usually things transfer in 3 ways: passive diffusion, incorporated within fat globules, or bound to proteins. With passive diffusion, it's according to concentration gradients from high concentration to low concentration until equilibrium happens. In this one, only free unbound drugs is available for diffusion through the membrane. And this is important to remember. And as this is the definition of diffusion, movement of molecules from high concentration to low concentration. And in breast milk, passage of drug into breast milk, there are 4 types of diffusion. Passive transport. The first passive transport is transcellular, where small unionized molecules, they pass unaided from high concentration to low concentration. And there is passive diffusion, where small ionized molecules and small proteins may enter the basal part of the cell from interstitial water through water-filled channels. And as you can see here in the picture, these channel proteins are water-filled channels. They allow substances to move through them from high concentration to low concentration into the cell. And these channel mediate, we call this type of passive diffusion, channel mediated facilitated diffusion, and it's facilitated by channel proteins. Some drugs use ion 4 diffusion, where polar substances penetrate by being bound to carrier proteins within the cell membrane. And you can see here, these are carrier proteins, and they bind substances and help them facilitate their diffusion into the lactocytes. And you can see here, we call this type of passive transport carrier-mediated facilitated diffusion. Now, another type of diffusion is intercellular, and it goes. This route avoids breast alveolar cell entirely. And if you look at this picture, this explains how large molecules enter human milk through these intercellular gaps. Another mechanism of drug transfer into breast milk is incorporated within fat globules. And if you look at the picture, this is endoplasmic reticulum, where fat globules are produced. And as they travel through the cytoplasm, they become bigger in size. Then they bulge from the cytoplasm, and they take with them part from the cell membrane. It remains sequestered within the fat globule membrane. And if you look closer at the fat globule membrane, the inside is full of fats, and the outside membrane has some proteins in it. So medication might be transferred into breast milk inside the globule entirely, or attached to the membrane, or attached to the proteins that are part of the membrane. The third mechanism is bound to proteins. Inside the vascular system, medication, they travel either within the plasma or attached to albumin. Those who are attached to albumin, usually they have difficulty moving across the bilayer from the capillary into the the mammary cells, because they are large molecules. Usually they don't pass. If you look at this picture, bound proteins, they move into breast milk mainly in this part here. In this slide, it shows the different how milk protein is formed and how it moves into the breast milk. The most important way is exocytosis of milk protein from secretory vesicle. And the second one are the milk proteins that are embedded within the membrane of the fat globules. And the third part is the plasma, maternal plasma protein that passes between the extracellular spaces. So if you go through this slide, you can find out that the main way proteins move, the medication move attached to proteins mainly through this one. There is also possibility in these two ways, but the main one is this, because these are being formed by this inside the cells, and these are being formed inside the cells. So in order for large molecules of proteins to go through the cell. They usually don't. They are usually passing through the extracellular spaces. Now, for the drug to pass into milk, it has to cross a bilayer membrane. The alveolar epithelial cell, which is the lactocytes, and the capillary cell wall here, and the interstitial spaces between them. And if you look at this picture, if the passages are closed, usually large molecules in the capillary, they don't pass into milk. Only those that are able to go through these bilayers are the ones that are going to end up in breast milk. But there's something special happening during early stages of lactation, colostral phase. The lactocytes are small in size, and there is a wide intercellular gap, allowing drugs and other products to move from maternal blood compartment to milk compartment in large amounts. Yeah, and you can see here the space is big between the lactocytes. Now, when lactocyte size increase, the spaces or gaps start closing, and it starts from 36 hours after delivery, and it ends by the 5th day up to 14th day postpartum. And usually this lactocyte size increase is aided by the drop in progesterone and the increased milk formation. And what we talked about, pathway 5, usually is open in early lactation, and then it closes, as you can see, during this period, 5th to 14th day postpartum is usually totally closed. It might reopen during pregnancy if the woman is lactating, breastfeeding, and she became pregnant later on, or she starts weaning and her breasts go through involution. And if there is inflammatory process, like when she has mastitis, during that, if you're taking care of women in these having this condition, you need to realize that a larger amount of medication can pass into their milk, and you need to keep that into consideration. Thank you for watching.